Well, can you hear me? The clock says 7 o'clock, so I think we ought to get started. First of all, let me just say I'm delighted to see all of you here. I was a little bit concerned that because the Forest Forward did not get printed in a paper form this week, that the word might not get distributed. So now I have much greater confidence than the fact that everybody looks at the computer for the schedule. Okay, so we want to welcome you to this Forest Speak program, and I'm going to turn over the program now to Dan Blazer, who's going to introduce our speaker.
Listen, I think we're in for a real treat tonight for someone who is just really quite amazing, and I'll try to introduce him as best I can. He's a native Tennessean coming from Western Tennessee. Went to undergraduate at UT in Knoxville, and then he went to medical school at Baylor, and then he came to Duke. And he spent a time in his residency and fellowship here at Duke in immunology, and then he went to NIH, where he studied with Anthony Fauci for 5 years. And so one little message I want to insert here is that this is not going to be a political talk. And so we don't know what's going to happen. And so I think Bart will basically tell you if you have any political questions, I don't know. I think as the rest of us are. He and Caroline, who's sitting right here, have been married for over 40 years. They have 3 children. They've been here at the Forest just a little over a year. Now, let me give you the real introduction for Bart himself. I'll start by saying I've been around Duke a long time. Bart Haynes is one of the 5 best academic medicine scientists at Duke now and has been for many, many years. And he's in some pretty elite company since we have 2 Nobel Prize winners in that group. He is the Frederick M. Haynes Professor of Medicine and Director of the Duke Vaccine Institute, and has been for some time. He's been at Duke for 47 years. He trained at Baylor, as I mentioned before. He is internationally recognized for his research, which focused on how the human immune system works and how to translate basic immunology into the development of vaccines for things such as HIV and COVID, et cetera. He's a member of all the major institutes, National Academy of Medicine, American Association of Arts and Sciences, and so forth. But I'll end up my introduction and give him plenty of time by just saying, not only is he extremely bright, he's extremely nice. And we are very fortunate to have Caroline and Bart here as residents at the Forest at Duke. Let's welcome Bart.
Thank you. Well, thank you, Margaret, for the invitation, and thank you, Dan, for that very nice introduction. I'm very grateful. Thank you all for coming tonight. So when Margaret asked me to talk about what I do at the Vaccine Institute and what is the Vaccine Institute, I tried to figure out what would be interesting. And I just decided to pick out 4 stories that have been interesting to me, and maybe they'll be interesting to you, about things that have happened over the last 40 years in the Vaccine Institute. So first I'm going to tell you, let's see. Go right? All right. So first I'm going to tell you what is the Vaccine Institute, and then I'm going to tell you story number 1 about babies with immune deficiencies. Then I'm going to tell you about story number 2 about what's going on with the HIV pandemic. And then I'll tell you about story number 3 about what we did in the COVID pandemic and how we survived that. And then 4th, I'll tell you about the bird flu epidemic in animals and the beginning of spillover into humans. And I don't know if that's going to be the next pandemic or not, but we're scared to death that it is. And so we'll talk about it, but I'm going to end up with some good news at the end, so don't worry. It's all going to be good. Okay, so these are our buildings. The Vaccine Institute is over on the Duke campus over here on the left at the Medical Sciences Research Building. We have that building. We outgrew that, and so I took a third of the institute and moved it out to the old GSK facility out in Research Triangle Park, that building there behind that wonderful fishing pond right there. And then when the crazy person sent anthrax to the senators and House of Representatives back in 2001, there's no better way to get Congress mobilized to vote on something than to send anthrax to them. And so they built 12 regional biocontainment laboratories, and one of them was at Duke, so we could safely do research on all kinds of bugs. And when I went to the Board of Trustees and I said, we'd really like to build a regional biocontainment lab here at Duke, the Board of Trustees members said, well, don't do that here. Don't bring all those bugs here. Just let them go somewhere else. And we said, well, I mean, that's fine, but actually they're already here in the microbiology laboratory over in Duke Hospital, and we don't have a safe place to do the work, and so all the work's being done unsafely right now. And that ended that discussion. So the other thing about regional biocontainment labs, and Wayne Koff knows this, is that whenever you try to build one of these, the community gets upset. So what I did was I got Bill Bell, the mayor, and all of the city mothers and fathers to come to Duke, and we said, we're going to apply for this before we had applied for it and wanted to get your buy-in, and it's going to help with public health responses in Durham. And finally, one of the city mothers, Livonia Ellis, raised her hand. She says, Bart, you're way overkill here. No one at Duke University has ever asked us to do anything before they did it. It's fine, go for it. So the mission of the Vaccine Institute is to develop vaccines and therapeutics against diseases of global importance while training the next generation of scientists. So what's our niche? Because that's what companies do. And we're an academic institute, but we're organized as a biotech company, but we don't have stockholders. We have stakeholders. Who are our stakeholders? Well, you are, taxpayers. And so we're heavily funded by the National Institutes of Health. We're heavily funded by the Department of Defense through this group called DARPA for pandemic preparedness and the Bill and Melinda Gates Foundation. We do work that's very difficult or may not be profitable for a company, but when we figure out how to do it, then we hand it off. And so good examples of the HIV vaccine. Nobody wants to make an HIV vaccine for money because we're going to have to give it away, basically. Same thing for making a vaccine and putting it on a shelf. What company wants to build a billion-dollar company around that where you don't even have a pandemic yet? But those are needed, as we'll hear. So we hand off things to pharma to get it into the public when we solve problems. So the three things that we're working on, or the four things we're working on the next generation of coronavirus vaccines, the next generation, or an HIV vaccine. We've been working on that for 35 years. A universal influenza vaccine, so you just have to get one shot and then that's it, and get another one maybe in 10 years. And then pandemic preparedness for anything that might happen, that might come out of animals, and be ready for the next epidemic. Now, this is the only slide about the immune system that I'm going to show, so hang with me. But this is one slide that you just need to hear these terms. So the immune system is our cells in our body that protects us from infectious diseases and cancer. It's comprised of two major types of cells called lymphocytes, and the lymphocytes can either be T cells or B cells. The T cells attack infected cells, so you get infected with influenza, it's T cells that clear them out. And also they control other immune cells and tell them what to do, so they're the orchestra leaders of the immune system. B cells, on the other hand, are the ones that make antibodies that neutralize the virus and kill the virus. Now, what we're going to talk about is children born with deficiencies of T cells and B cells, and that disease is called severe combined immune deficiency disease. And we're going to talk about children born without a thymus gland, which is the gland that makes the T cells. And they have no immune system because the T cells are not around to tell the B cells what to do. So let's start on story number one. These are two patients that changed my career. And these are two wonderful teachers in their older ages, Bill Shearer and Marianne South. They were at Baylor. They're two of the most wonderful doctors I've ever met. And this is a baby named David Vetter, who was born in September 1971. Has anybody heard of David Vetter? Anybody heard of the boy in the bubble? Yeah, okay, there you go. So this is David Vetter, and Marianne South and Bill Shearer studied this boy in utero and knew that he was going to be born with no immune system. And so what they did was, when he was born, they put him in this containment area so that no germs could get into the bubble. And this is little David when he was about 1 year old in 1972, when I was in medical school and doing my rotation at Texas Children's Hospital. And who was my attending but Bill Shearer and Mary Ann South. And they took me into this room and introduced me to little David. And it's literally was the most profound thing up to my life at that time that I had seen, this concept of no immune system, what have you. Well, the story went on, and David grew up in the bubble. This is Bill now, long after I'd left Duke in 1979. And then here's David. NASA made him a spacesuit so he could get out and walk around. This is all from the internet, so this is not pictures that I took. And then here's David in 1984, just before he died, when there was a group at Harvard that thought that they had figured out how to do bone marrow transplants for severe combined immune deficiency disease, and they tried it, and it didn't work. What they gave him were cells that acted back on him, and he died. And it was very tragic. And the irony of this is that if he had waited 5 years and brought him to Duke, Rebecca Buckley, who many of you probably know, and 5 years later figured out how to do this, and he would have lived. And she used her technique and saved literally tens, if not hundreds, of babies. Now, if you want to see a movie, and John Travolta's The Boy in the Bubble, this is a movie that was made in 1976 about David Vedder. It's highly stylized and made for Hollywood. So you fast forward to 1980, and I came to Duke back from my 5 years at NIH with Professor Fauci. And I arrived from the NIH, and I was studying the human thymus. That's this gland that sits up right on top of the heart. It makes all the T cells that go around the body and tell everybody else in the immune system what to do, and creates a normal immune system. And so children born without a thymus, therefore, have no functioning immune system. They die in the first year of life, just like severe combined immune deficiency disease. So we were trying to learn how to do thymus transplantation, and the way we were doing it, we were taking little bitty pieces of human thymus and taking it with a needle and sticking it under the lining, outside lining of a mouse kidney, where it would grow and get nutrients, and to see if we could get it to grow. And so this is over on the left-hand side is a real mouse kidney, and the circle drawn around it is, you can see a little white thing in the middle. That's the human thymus growing under the kidney capsule of the kidney. And if you cut it in half and you look at it up there, the thing that's circled is the human thymus growing on top of the rest of this thing, which is a kidney. And you just have to take my word for it. All those blue dots mean that that's a normal thymus, and the thing is growing. So I showed that picture at a conference over at Duke, and Louise Market, who at that time is a young pediatric infectious disease physician scientist, she just finished her MD and PhD at Duke, and she said, if you can get the human thymus to grow in a mouse, I'm sure you can get it to grow in my babies born without a thymus. And so we took that on, and from 1985 to 1990, and there's a picture, a drawing of the normal human thymus, and for a baby born with congenital heart disease, the thymus is about half the size of your hand, sitting right over the heart, and a baby's heart is about the size of a walnut. So that thing has to be cut away by the surgeons so that they can get to the baby's heart and fix a hole in the heart or whatever's wrong with it. And so they, instead of throwing the tissue away, we went to an institutional review board protocol, and we got the tissue and started growing it in the laboratory in such a way that it wouldn't harm the baby. Now, the metaphor is that every child needs a school to go to learn. And think of it this way, every T cell, young T cell, needs a thymus to go to learn how to be an adult functioning T cell. So in a sense, what we were trying to do is learn how to grow the thymus from another child and get rid of all the cells in it that would hurt somebody, that you would put that in there, but just put the schoolhouse by itself and let the babies, young T cells, come to this thing wherever they are by going around in the vasculature and stick there and learn how to become mature T cells. And so what we did was we figured out how to grow these things in tissue culture. And then along came about 1990, a young girl, about 3 months old, who was born without a thymus. and she was very sick. And so we put 20 or 30 pieces, and instead of opening her chest and putting it where it normally should be, we just said, these cells are going to figure out where home is. And we just put them in the thigh and in the muscle of the thigh just to make it easy to do. Lo and behold, it worked. So this is the first paper of successful formation of this thymus graft transplanted from cultured postnatal thymus. And so at that time, doing work on fetal thymus and fetal tissue was not kosher. And so we figured out that you could do this with thymus taken from a cardiovascular surgery, corrective surgery. So this is the first successful cultured thymic transplant, developed into mature T cells. And this patient is now 36 years old and doing fine. And Louise Markert, over the next 35 years, Louise dedicated her career to saving all the children in the world who could come to Duke with this, over 80 or so at the last count. But more importantly, she got thymic transplantation approved by the FDA, so it could be done anywhere. And more importantly, she set up a unit and taught the people in Europe to do it, so that it had 2 places where babies could come from all over the world. So that's the story number 1, and let's go to story number 2. And this is the pandemic that changed my career, and that's acquired immune deficiency syndrome, or AIDS. As background, in 1981, just after I arrived here at Duke, AIDS appeared in San Francisco and New York City and spread rapidly across the United States. I had just moved to Duke from the NIH, and Bob Gallo at the NIH called in 1981 and wanted me to move back to NIH to work on this new disease. Well, I didn't want to go. I just come here and set up shop. And so I stayed at Duke, but I went to the NIH every month, and I added AIDS to work in my own laboratory. Now, the way the conversation really went, I don't know if you've read about Bob Gallo, but he's a very, very strong personality. He basically said, you have to do this. You don't have a choice. The government has paid for you to become an infectious disease doctor, because I was on a training grant. I had already done work. They thought it was a retrovirus, which is the kind of virus it turned out to be. We'd already done work on that. And he said, you don't have a choice. You got to do it. And I said, okay, well, I'll travel and come up and go back and forth. So we did that. And so this is a series of slides that I dug out from deep in the internet from back in 1980 from the CDC. And this is every dot is 20 cases. This is where they were over in San Francisco and Los Angeles, and then up there in New York back in February of '83. And 2 years later, it started to spread. 4 years later, 100,000 cases. And 4 years later, 330,000 cases. And about 1990, 93, we knew we were in really big trouble here. And so what we did here, the NIH, the National Cancer Institute, had built a building for cancer viruses called, and I saw Harold Erickson here, and Harold knows, and Jerry Lazarus knows, and several other people know about these buildings that are over at Duke, and Jeff, and that to isolate cancer viruses. And so we fired up this thing called a Blickman line there, which nowadays nobody uses these. You walk in rooms and you have your own system to keep air away from you from whatever the organism is. But here, you kept air away from you by putting your arms in these things and working inside these metal containers. And this is called a Blickman line. But you could do tissue culture and microscopy and a variety of things. There's Danny Bolognese over there that I work with for many, many years on this problem. Danny's a resident here. I don't know if he lives here if he's here and physically here now, didn't see him, but Danny is a resident and is a giant in this area. And I was fortunate to be able to work with him. And both of us, we would go up to the Gallo lab meeting. And my job, there was a new group called the people who got blood products because they had clotting problems called hemophilia. And they were a new group at about 1983 that were emerging with this new immune deficiency disease. And so my job was to organize all of these studies on this particular group of folks. And so we did that, and we analyzed serum from hemophilia individuals in that Blickman line over a couple of hundred individuals. We sent samples to the Gallo lab, and they isolated several isolates, but one in particular that proved important. And we found a lot of serum that he used as a diagnostic reagent for whether other people were infected. And Bob Gallo's work with a series of 4 papers back in 1984, a year after the French discovered the virus, proved that it was the cause of AIDS. And so soon after that, Danny and I started to work on a vaccine. We thought it would take about a year. Wayne Koff was at the NIH, who's here today, and he knows all this whole story of the NIH helping to fund all of these candidates for vaccines. And this was the first Duke vaccine candidate, and we partnered with Wyeth Lederle, and they made it. And we thought we'd done it, it's fine. And it did what it was supposed to do in people, except against the wrong viruses. We were using cultured virus and not the right kind of virus. And that set off now 35 years of failures. So this is Albert Sabin. Albert Sabin is holding in his hand the cube of sugar into which many of you maybe have gotten the polio vaccine, put it on your tongue, and this was an attenuated or weakened polio virus. And this was in about 1990. This photograph was taken. You see he's got his cane there. And in 1990, a good friend of his named Sheldon Wolf, who's one of my mentors at the NIH, had a symposium and invited Albert to it. And Albert couldn't get around. And so I was a young guy at the time, one of the younger people at this conference. And so Sheldon said, you stick with Albert Sabin and help him go wherever he needs to go. So I got to sit next to Albert. Every time he got up to talk, I got up to go with him and eat and all this kind of stuff. And so we struck up a friendship. And so after that, we started writing. And here's a little note up there. He says, Dear Dr. Haynes, he was just the nicest man. He was just unbelievably friendly and courteous, tough as nails scientifically. And he said, Dear Dr. Haynes, many thanks for your kind letter of October 25th and your accompanying paper, which you just, your important paper that in the Journal of Immunology. I have just, quote, read it with great interest. Now, I don't know what that means, but you'll see in a minute. And I do wish you success in your future work. Kindest personal regards, Albert Sabin. And then every month or so, I'd get a paper like this that would be underlined, notes in the side of it. And the top of this one says, Dr. Haynes, just in case you may have missed this, best wishes, Albert Sabin. And then 2 years later, and he firmly believed that it was going to be incredible. He was an unbelievably smart person. It's going to be incredibly difficult to make an AIDS vaccine because of the biology of it and all this. And basically, the virus coats itself with its own with our own molecules and fools us in saying you shouldn't respond to this thing, different than any other bug that we've tried to make a vaccine with. And he knew this all back in the early 1990s and what have you. And so 2 years later, he wrote this paper, Improbability of Effective Vaccination Against Human Immunodeficiency Virus because of its intracellular transmission. And he tried and tried and tried to convince me that I was wasting my time to do this, and I did. I wasted 35 years. But then Bill Gates and Tony Fauci got together in 2005 and said, the field is a total mess. Everybody in the field has all these vaccine candidates, and nobody will share them. Nobody will compare them. We got to quit doing all that. So Tony Fauci went to Bill Clinton. Bill Gates went to Bill Gates, and and Fauci got $340 million from Bill Clinton for AIDS vaccine work, and Bill chimed in a similar amount. And the government gave their $340 million over 7 years to one place, and that was us at Duke. And Bill Gates divided his up into about 10 centers. But the point was you had to work together, you had to talk, you can't compete, you've got to share data, and you've got to compare everything you're doing and quit wasting time. So it was a total sea change, and Richard Klausner at that time was a Duke graduate, and I don't know if any of you from Duke knew him, but he's probably one of the smartest medical students ever to come through Duke. And he was the scientific director for the Gates Foundation. He got everybody together, and we put together this paper basically outlining what needed to be done. And so out of that came 3 grants. The first Chavi grant that came to Duke, and then the next 2 grants that one came to Duke and one went to Scripps out in the other coast. And that's a group of the people that have worked together with us for now 19 years on this particular project. And without going into a lot of detail, we brought some new technology into it back in 2016. And this guy named Drew Weissman came to me and he said, look, I've got this new technology and nobody will listen to me. It's called modified messenger RNA. And it's going to work. But I can't get enough monkey to do a monkey study to show that it works. And I said, well, great, let's try it. And I said, do you have HIV mRNA made? And he said, no, I haven't done that yet. All I've got is Zika. So I called up the NIH and I said, look, I've got this money for HIV, but we want to test it in a monkey trial to test this technology, and it's on Zika. Can I use HIV money to make a Zika vaccine? And they said, absolutely, go for it, do it. And so we did it, and it turned out that the mRNA Zika vaccine in monkeys was 100% protective and 100% safe in the monkeys. This was in 2016. We wrote it up, published in Nature, and 5 years later, it won this award from the Spanish government, awarded by the King of Spain for this as the first mRNA vaccine that had worked. And Drew Weissman and Caitlin Carrico had, back in 2005, discovered how to do this. And they won they're the 2 that won the Nobel Prize in 2023 for this. And so when all of the work started coming out for mRNA and getting an mRNA vaccine in 11 months and all that, we knew, oh, this is going to work. There's no question about it, because it had already worked in monkeys. So let me just take a couple of slides and just explain to you how messenger RNA works. So messenger RNA is on the left-hand side. There is the template. It's genetic material. And it's the template on which proteins are made. And so one of the secrets was to take some lipids that makes it like a little bitty cell, or actually a little bitty virus, and coats the mRNA with lipids, and that protects the lipids and also allows it to get into our cells once it's injected into the arm. And so what the vaccine is that the RNA in a lipid nanoparticle is injected into the arm of the vaccinee, and then once it gets in our muscle or whatever cell it gets into, then it encodes a protein, and the protein is made. So most vaccines, you just give the protein, and that's it, and the immune system works on it. But here you give the mRNA, it makes the protein in the body, and the immune system recognizes the protein and makes a protective neutralizing antibody. And one of the many advantages of this is that you can make this vaccine in a week, and I'll show you an example of that. So right now we have 2 ways, and we have 2 HIV vaccine candidates that are starting to work after 35 years. And one vaccine is a protein that makes it look like the vaccine looks like a virus. And it's a synthetic protein made by Kevin Saunders over there that works with me in the Vaccine Institute. And it looks like a virus, and it fools the immune system to respond. And it says, don't worry about it. You're going to be okay. Just make this, and the antibodies are coming out. So that's really cool. And then Saunders, working with Drew Weissman, has figured out how to make this as a messenger RNA. And so this is the gene that gets into the body, as I said, and makes the protein. And it too fools the immune system to respond. So we think we're on a positive path. And to Albert Sabin's credit, that paper he wrote, the last sentence in that paper says, you're going to have to do something different. You're going to have to find another immunologic technique and technology, because what you're doing now is not working. And he was absolutely right in 1992. I wished I'd listened to him. So now we're moving on to story number 3. This is another pandemic that changed my career, and that's, of course, COVID-19. So what we did when, in January of 2020, when the virus, it was obvious this was going to be something we were going to have to deal with and we couldn't ignore, the president of Duke University came to us and they said, it looks like we're going to have to shut down fairly soon. And so indeed, in March 21st, Duke University shut down, closed all the laboratories, and he said, before we do that, I want you, the Vaccine Institute, to develop a test that you can test everybody at Duke, the faculty and the staff, and keep us open and know who's infected and who's not. And so we turned, this is the inside of the Vaccine Institute, that building there, Medical Sciences Research Building. It's filled full of all these boxes full of vials and all this stuff to do tests on. And we did 1 million laboratory tests for called polymerase chain reaction, the genetic test, the definitive test of whether people are infected. We did over 1 million tests over a 3-year period of time. And all the students had to come in. It was incredible. The whole community just fell in line. They came in 2-3 times a week, got tested. If they were positive, they went off to their dorms. They quarantined for 5 days. Same with the faculty. But everybody, the thing stayed open and the classes occurred. And it cost Duke multiple millions of dollars to do this, but they would have lost 10 times more money than that, and time for the students, et cetera. So this was an article in the Los Angeles Times. Duke University schools the country on how to stay open during the COVID-19 pandemic. It was also written up by the CDC, and unfortunately it just wasn't replicated in maybe 2 or 3 other places. That was about it. From a scientific standpoint, we knew that BioNTech and Moderna were making mRNA vaccines. We knew they were going to work. But we also knew that this virus, from our work with HIV, which is one of the fastest evolving life forms on Earth, this virus is a similar virus. It's an RNA virus. We knew this thing was going to evolve and evolve quickly and escape the initial immune response. So we started working on the next generation vaccines. And this is an article, and this is the vaccine here over on the right-hand side, where we took a synthetic piece of a bacterium in that gray thing. It looks like a little bitty, tiny little soccer ball, and it's about the size of a virus. And that red and blue thing on the top of it is just a little piece of the COVID virus, the SARS-CoV-2 virus, to which the neutralizing antibodies bind. And this thing worked really, really well for inducing quite broad responses to various coronaviruses. And so we have a manufacturing facility. I see Harold Erickson came in, and Harold Erickson taught us how to do these electron micrographs over there on the left-hand side. And that's the artificial version. The computerized version has the blue and the red on it on the right-hand side. And then those pictures there are the real thing of the thing made by our manufacturing facility over at Duke. This was done on November 20th of this year. It's made. And there's a bottle of it. Well, clinical trial will start January of 2025. Now, we've already gone ahead and made a broader, even more expansive pan-coronavirus vaccine. It's made the same way with the same technology, but instead of having just the blue on there like the other one did, it's got a different species of coronavirus in red and a different one in green, and gets a much broader response because it hits all 3 of these kinds of viruses. Now, this is the last slide on the coronavirus pandemic. It just shows what's happening now. And the orange peaks are cases, in the instance of cases, and the blue are deaths every week. And so I don't know if you can see the lines there, but every line there is a week, and it's the number of deaths. And what the virus is doing, the virus has said, yeah, you've got a vaccine, and the vaccine prevents serious disease, but it doesn't prevent transmission. And so the epidemic is continuing in these peaks. And what the peaks represent are evolved viruses that change and escape the neutralizing antibodies that we've induced. So there's Delta over there and Omicron. And fortunately, with JN1 and all these new ones that we don't really care about because they're not causing increased death and hospitalizations. But the vaccines now are designed to take care of these new ones. The new vaccines that are coming are going to be vaccines that you can prevent transmission. And that's transmission because where we get the virus, and that's up in the nose and the upper respiratory tract. As we're trying to figure out if we can end this pandemic so we can prevent that those orange spikes by ending transmission. So those are the new vaccines that everybody's working on. Now, the last story is a new bug that we're getting ready for, and that I really hope it's not going to change my career. And that's the hot topic of avian influenza. Have you all heard of avian influenza and you've been reading about it? And yeah. So what's happened is, over there on the left, it says birds have infected. So the natural host of avian influenza is birds. And birds all over the world are the host, and usually the infection doesn't break out and go all over the world. Well, for some reason, probably traffic and shipping and all kinds of things, birds have infected many animals, including many herds of milk cattle around the world and in the United States. And the red dots there and the animals, and that milk cow's udder represents the influenza avian virus, and it's live virus, and it goes into the bucket of milk. And the people becoming infected are the milk workers. And so going over to, it says persistence in raw milk. So you don't want to be drinking raw milk. About a third of all the milk in the United States is contaminated with H5N1. But fortunately, the good news is pasteurization takes care of it. And cheese made from it takes care of it. And it's a debate about whether unpasteurized cheese made from raw milk, because of the acids and stuff, that may take care of it, but I don't know that. And that's what this article is about. And then the question is, if you get a lot of this stuff over there, then some of the humans are becoming infected, but thus far there hasn't been a large pandemic, as we'll talk about. So to infect humans, the biology of this whole thing is that bird flu has to mutate at least 4 times. It's got to change. It's an RNA virus too, and it's mutating just not as fast as HIV and then even slower as SARS-CoV-2 for COVID, and even slower as bird flu, but it's still mutating. So bird flu has to mutate at least 4 times. First, it's to jump from birds to another animal, like cows. It's got to mutate. Number 2 is to learn to jump to humans from whichever animal. Number 3, to learn to cause disease in humans. And number 4, to go from one human to another human. Now, thus far, it's probably more now. This was in November. 52 humans infected in the United States. They're only getting mild symptoms, conjunctivitis, and not serious influenza, pneumonia. But they're not seriously ill. That's good news. Then this was an article in the New York Times Saturday, 3 or 4 weeks ago, from Toronto. It says a teenager was hospitalized in critical condition after contracting bird flu. Health officials were investigating how the teenager came down, because the teenager had nothing to do with poultry farms, nothing to do with cows. Implication was it got it from another human. Then the second bar in the middle, it shows sort of what happens when epidemics occur. And so about a week before this teenager went into the hospital, they went to the emergency room with respiratory symptoms, had pink eye, fever, and a cough. The teenager was sent home because nobody thought that the teenager was very sick. The teenager was actually quite sick, did not go to school, fortunately. And then the good news is 36 people who came into contact with the young person all tested negative. They were all offered a preventive course of antiviral medicine. We'll talk about that in a minute, another piece of good news. At last, I've read about this individual. They're still extremely sick on a ventilator. So 4 days ago, this paper came out in Science magazine. And the point of this, and that you may have read about it because people made a big hullabaloo about it, they said that they took the cow bird flu, that strain, and they made a single mutation in it and showed that when you made a single mutation in it, it now bound to the human receptor. And they couldn't make a virus that way because that's illegal. That's called gain-of-function research. But you can take the artificial protein and show that. And what this paper says is that one more change, mutation for the Texas, it's called H5N1, has to go for easy human-to-human spread. And that really got a lot of people's attention. So the notion is that, and this is what the press and scientists and everybody's been saying for now 6 months or more, is that we're in for it. We better prepare for it. Got to do something. So here's the level of, as of November 18th, there have been reports to Department of Agriculture of almost 11,000 wild birds infected, 108 million poultry and farms either sacrificed or known to be infected, 48 states with outbreaks in poultry, 15 states with outbreaks in dairy cows, and over 500 dairy herds. And this is the tip of the iceberg because none of the folks that run these farms want people to come in and test. The CDC has to be invited by the state. There's no federal mandating what the CDC can do. And so their program now is to only go when there's an outbreak and test, as opposed to going into all the herds and all the places and testing before outbreaks occur. So that's why people are complaining the United States is not doing enough. So what's happened over the past 8 months or so in other parts of the world is astounding, in that bird flu has infected a lot of other types of animals. So seals in South America. Seal colonies in South America 1 year after a deadly outbreak kill more than 17,000 elephant seals and 97% of the pups. In Washington, there was an outbreak in 2023 where half of a large breeding colony of seabirds, Caspian terns, died from highly pathogenic avian influenza. And there was recently an article talking about animal conservationists saying we're really worried that part of our animal species are going to collapse because of this spread of this virus in animals around the world. So let's end up here on some good news. We have drugs. When the COVID pandemic started, we had no drugs. Now, we have 4 drugs for pathogenic avian influenza. One is oseltamivir, which is recommended to start very early on with flu symptoms. Another, peramivir, can be used to treat bird flu, as can another called zanamivir. And then one for older individuals with immune system problems. You can combine this one, baloxavir, with oseltamivir for immunocompromised patients. So we're not starting from zero here. That's good news. Now, what about vaccines? Well, I've mentioned that we've made our vaccines at Duke, and 10 years ago we built a manufacturing facility. The NIH came to Duke and said, "That is the dumbest thing you've ever done, Haynes. Why didn't you do that?" Literally, they said that. And so we hung in there. We made our first vaccine. 2 years later, the NIH came back and he said, "Haynes, that's the most brilliant thing you've ever done. That's wonderful." So we have this fantastic unit and these wonderful people over there that lead this. And for mRNA, we can make mRNAs and we can make proteins. And for mRNAs, we're the only group in the world that's published what we do. We wrote that paper right there. Now it's a Science Citation Classic. That just means a lot of people cite it because we're the only ones tell people how to do it. And we've helped people in South Africa, in Thailand, and maybe 5 or 6 places in the United States Army and places all over the United States learn how to make mRNA vaccines. And so I guess it was June 2024, we received a call from the NIH. And the call was from the director of NIH and the people, Wayne, from the Division of AIDS. And they said, "You have a GMP facility that's making an AIDS vaccine. We want you to stop doing everything you're doing. We want you to make 2 H5N1 vaccines, and we want you to do it quickly because the White House just called and we need more vaccines to put on the stockpile." So we stopped everything that we were doing, and over a 4-month period of time, because I mentioned earlier, one of the benefits of mRNA vaccines is that you can make them very quickly as opposed to proteins that take years to make. And so we made these 2 vaccines. One is called H5N1 and the other is H5N8. H5N8 is just a variety that's over in Southeast Asia, and H5N1 is like the one in Texas. And the clinical trials on these are, again, going to start sometime the 1st of January at Duke in our clinical trials unit. And so the final slide is this is the real good news. We have vaccines already before this pandemic has really taken hold. We have 3 vaccines the government has already stockpiled. one made by a company called Sanofi Pasteur, one by a company called Seqirus. Now, Seqirus is one of the companies that makes most of the vaccines that we take every fall. And then the third one is in a company in Canada that the United States has bought a bunch of vaccines from. And those three are all traditional, old school, grow up the virus in eggs, kill it, put it in a vial, and that's what we get every fall. Then there are the two messenger RNA vaccines that I just mentioned that are going into clinical trials for both safety and to make sure that they do what they're supposed to do. And then the government will take them and give it to a company and grow up 200 million vials or whatever they grow up and put it on the shelf. So it takes a village to do all this work. This is about half of the 300 people we have in the Vaccine Institute doing all this incredible work. What a team. The people are incredible, and they're all just as smart as they can be, and they're all committed to what they're doing. It all takes an international village to do this work. These are all of our collaborators all over the world that we work with on various projects and collaborate, share information, and do all we can to protect the public. And that's pretty much it. I'll be glad to take some questions if anyone has any questions.
Thank you.
You're welcome.
I've been involved as a volunteer in research for a long time because of a bargain I made a long time ago with Providence. And I want to know, I'm 85, I've just finished the 6th year of the senior flu vaccine at Duke. I was in the COVID test. Should I continue volunteering, especially with the avian flu?
Should you continue to volunteer for clinical trials? Well, let me just say, I am an MD. I quit practicing about 20 years ago. And so I'm probably not the best person to give you medical advice. But I am, how old did you say you were? 85? Okay, well, I can just tell you my own personal experience. I'm 77, and my wife is much, much younger. And we both volunteered for a clinical trial and gave blood and all this 2 weeks ago. And so we're doing that. Whether you should or not, I don't know, but I would leave that to you and your physician. But blessings on you. Hey, Jeb.
Hey, Bart. I'll just share a small world story that you may or may not remember. So I was recruited to Duke by Danny Bolognese, who Bart mentioned. Danny headed up a group in the Department of Surgery called the Surgical Virology Unit. We were all immunologists and virologists working with mouse retroviruses, which was very similar to HIV when it was eventually discovered.
Yeah.
And I was a member of the Duke Comprehensive Cancer Center, and one of my responsibilities was to run the Cancer Center seminar series. And somebody had recommended this guy at NIH to me, who would be a good speaker, and I contacted you, and you came down, gave a terrific lecture, very much impressed Bill Shingleton, who was director of the Cancer Center at the time, and Danny, Bill Yaklik. Eventually, I think coming out of that, you were recruited by Duke to come. So I consider myself partly responsible for your coming here, and you're
outstanding
at the forest.
Thank you very much.
Absolutely.
We never left, so it's great.
It's a pleasure, and you've done fabulous stuff.
Thank you so much. Thank you so much. Hey, yes, Jerry. Oh,
Bart, you've dramatically demonstrated the importance of vaccines and immunological responses to epidemic diseases. You've demonstrated quite dramatically the utility of vaccines in the control of disease. You and I and many people in this room are probably alive and well because of the polio vaccines.
yes.
The issues in the future are somewhat concerning. what is the vaccine community doing to develop programs to persuade the United States, the people of the United States, that to diminish vaccine research is a tremendous problem?
Yeah, well, that is just a terrific question. The honest answer is, Jerry, we're in a no information zone right now. We don't know what's going to happen. I don't know what's going to happen. I don't know if things are not going to change that much or if things are going to change dramatically on January 21st. I just don't know. The way, again, all I can really do is to give you my own personal, what I've done the last week. I begun thinking about how the Vaccine Institute can better message what we do, giving talks like this to try to say, this is what the government has done. That's the truth. That's what's stockpiled. So how do we message what we do better? I've started to read a lot more about what other people are saying that may not agree with how I think, to try to understand where they're coming from so that I can more articulately engage in conversations. From a practical standpoint, we are working hard to explore the possibilities of funding from a variety of sources, not only the federal government, but other places. So those are the main things we're doing, and we're just trying to bide our time and keep trying to spread goodness and light while we wait and see what's going to happen.
You know, it seems to me that you've been very prescient with the possible influence of avian flu. You know, here you are and you've made major changes in anticipation of an epidemic which sadly may very well occur. That sort of forethought with regard to the possibilities in your elevated position, what can be done to put together a team approach of immunology, vaccinology, and so forth to prepare to do something with regard to the possibility that this may come to a screeching halt.
I think the question is that, Haynes, you just said you got all these people together. Why don't you do something about what something bad might happen, and that all this vaccine work may come to a screeching halt? And I don't have, Jerry, I really don't have a profound answer to that. We just got to see what actually materializes and go from there. There are a lot of people talking about it, but we need to see exactly what materializes. I think that's the most honest thing I can say. There's a question back there in the back.
Bart. Oh, okay.
Is that Howard?
Bart.
I recognize Howard, yeah.
Oh, okay. Bart, the avian influenza virus hasn't mutated to the degree that there is human-to-human transmission to any
Right.
great extent.
Right.
So the vaccines that are being produced now presumably wouldn't necessarily be effective against that mutation in the future, or am I mistaken?
Well, this is a really good question. And so what's being done is the two vaccines we made have been, those viruses have been studied, and they are sensitive to the antibodies that are being made in the cows and the few people that are being infected with this thing in Texas. So, it's not the mutations that you have to target, it's the conserved regions on the pieces of the virus that the antibodies have to bind to neutralize, and they're generally conserved. And so those tests are being done to make sure that whatever vaccines are put on the shelf by the government will be useful. And it's always an estimate because this virus does mutate and change in response to antibodies. So it's a great question.
And my last question, as an oncologist, I've always been interested in therapeutic cancer vaccines. I know this isn't necessarily work that's being done at Duke, but what's your prediction as to our having a therapeutic cancer vaccine in the next 5 to 10 years?
Well, as you just said, this is not my field, but I will make the point that the technology that allowed the COVID response to be so quick was developed over the previous 20 years. And I gave one example of it with Drew Weissman and the messenger RNA 10 years ago. All of that work was done and money paid for to try to do all these, to make the HIV vaccine and come up with all these failures for the HIV vaccine. But all this technology allowed all of this work to go with lightning speed, et cetera. The same thing can be said for work for cancer vaccines. Hot, very important area of particularly using messenger RNA. We're actually getting ready to collaborate with institutions up north to make mRNA cancer vaccines against cancer cells, et cetera. So it's a big deal.
Okay,
Dan?
are we on? Yeah, we are on.
Yes, sir.
The thought occurred to me that to a certain extent, this is a financing problem. And if you didn't get financing from Group A, is it conceivable that there's enough money available from Groups B and C that could continue the work, to continue to have the work funded?
Oh, absolutely. I think there are many different possibilities, and whenever wrinkles occur in the norm, it stimulates new and creative thinking about other avenues, both scientific and, as you're bringing up, financing. So I would say so. And there are other people here probably who have opinions about what might be available through other avenues other than the federal government. We'll just have to see. Yes, sir.
Not getting
I'm going to defer that to your own physician. As I said, I haven't practiced in 20 years. I can only tell you about my own 77-year-old self when I got it. Any other questions? Margaret has a, oh, yes, sir.
Thank you so much. This has been great. I have more or less a practical question. This virus that goes from the bird to the cow, and then possibly from the cow to the human, it seems like it goes through the milk of the cow. Where is actually the virus repository within the cow and then in the human when it gets there?
Yeah. Well, what this virus does is it binds, it uses a part of the virus that binds to sugars on our cells. And there are different sugars in the eye and sugars in the lung. And that's why most of those 50 people are just getting conjunctivitis, because it can bind to the sugars in the eye but can't bind to the sugars in the lung. And so it's cells on the lining of our organs called epithelial cells that the flu virus gets into, hangs around. It's also, yeah, secreted into the milk, right? Mm-hmm. Yes, ma'am. Yeah, the question is, how do the cows get it from the birds? I actually don't know the answer to that. I assume it's through the environment and bird poop and all that. I don't know the answer to that fully. Any other questions? Yes, ma'am.
You described being asked, I believe, by the NIH to immediately stop what you were doing in producing HIV, AIDS virus, or vaccines?
Well, we were making AIDS vaccines, we were making coronavirus vaccines, we were making other flu vaccines.
And it's wonderful to have this repository of bird flu vaccine, and yet it must be painful to stop progress on the AIDS work that you spent so many years on. Is that true?
Well, I wouldn't say it was painful. I was saying it was annoying because we didn't really stop. We just, okay, we've got 6 things we have to make, and it was like moving blocks around. And we said, we want to do whatever we can do and get this thing out by November without messing up everything else. And so we did a fairly good job. We probably lost 2 months, and that's okay for this cause. We believed in this cause, and it didn't take a lot to convince us this was really important. We were really worried about this bug, so we lost 2 months. No, it didn't. Any other questions? Well, thank you all for coming out tonight. Wow. It's just been a lot of fun to talk to you all. Thank you.
You are right at the cutting edge of this moment.
Oh, I can take your phone. Yeah, I can. Of course. Here you go. Of course, you're welcome.